I am very excited about us hosting this 4th annual lecture as part of our 30th annual Alumni Weekend, which is terrific. And before introducing our distinguished lecturer, I do want to take a moment to acknowledge Hal and Carol Kohn for their generosity in establishing this esteemed lectureship. Hal is a professor emeritus at the school. He was recruited in 1999 to UNC to lead the transformation of what is now the Division of Chemical Biology and Medicinal Chemistry. He also served as the first director of the Campbell Faculty Mentoring Program. And in 2018, Hal was the recipient of the school's Distinguished Service Award in recognition of his exceptional accomplishments and contributions to the school and to the profession. In establishing the distinguished lecture, Hal and Carol hope to honor the contributions of our alumni, to recognize faculty and staff who have empowered alumni achievements, and also to inspire our students. So thank you very much for bringing our community together through this lectureship. We appreciate you very much. And now I am delighted to introduce this year's Kohn Distinguished Alumni Lecturer, Dr. Kellie Schoolar-Reynolds. Dr. Schoolar-Reynolds is the director of the Division of Infectious Disease Pharmacology in the Office of Clinical Pharmacology at the FDA. So she is a big deal. She completed her fellowship in clinical pharmacokinetics and drug development with the school in 1994, after which she began her career with the FDA. Her work with the FDA began during a pivotal phase of HIV development, which allowed her to make crucial contributions to developing therapeutics to combat this life-threatening disease. Through this work and continued policy development, regulatory research, and stakeholder engagement, Dr. Schoolar-Reynolds has found the keys to staying engaged and energized with a career of nearly 30 years at the FDA. So I am honored very much to have Kellie back with us on campus, and I invite her to come up for the lecture.
Thank you very much for the introduction, and thank you so much for the invitation to do this lecture. I really appreciate the inspiration behind the lecture. Celebrating the contributions of the various alumni of UNC School of Pharmacy, the various programs, whether it's the PharmD, PhD, the fellowship program. And even in my day-to-day work, I see the impact of individuals who are alumni of UNC. And I've worked in the federal government for 30 years. Not many people work in the federal government for 30 years and think that they have the best job in the world. But one of the reasons I wanted to come here today, and I was so excited about it, was so I can tell you why my job is so fantastic. I'll also point out that my division has 20 individuals, including myself right now, and 4 of us are alumni of various UNC School of Pharmacy programs. So there's one who is a T32 UNC Duke alumni. We have one PharmD graduate. We have 2 PhD graduates. So having 4 from out of 20 is a lot, and we're actually getting ready to hire someone else. So we're in the progress of even increasing our numbers. So that is very exciting. So it's been a long time since I've been here. I was here from 1992 to 1994. I worked mainly with Dr. Kim Brower. I know she couldn't be here today, so I did get a chance to chat with her about that, about the time that I spent here. And I've watched the progress of the school over time, and it's been amazing. So I follow you on Twitter, on LinkedIn, and I get to see what's happening over time. So I'm going to start by talking about my career journey, going back to even before I was in college. So my outline for today, I'll talk about my journey. And then I'll tell you a little bit about the FDA, because I know for the most part, it's just this black box. No one knows what's going on at FDA, what the structure of the FDA is. I will highlight a few of the impacts that I've seen of clinical pharmacology, clinical pharmacy at the FDA over the past 30 years. And then I'll step back and talk about, at a higher level, what makes a rewarding career, and how what I have experienced has really been a rewarding career that you could apply really in any setting. But I'll start with my journey. So going back to the formative days in school, high school, and before that, I was always interested in math and science and healthcare. I was one of those kids who said, oh, I want to be a doctor when I grow up, knowing that I was not going to be a doctor when I grew up. But it sounded good. But I knew that there had to be math and science involved. Then I went to Virginia Tech. I had received a BS in biochemistry, not knowing what I was going to do with that. But I did know that it was an area of interest. And towards the end of my time at Virginia Tech, there was like this list of things that you can do with a degree in biochemistry. And one of them was go to pharmacy school. So, okay, I'll go to pharmacy school. So then I went to Virginia Commonwealth University. I started out in the BS program. This was during the time where it was transitioning from BS to PharmD. So I switched in the middle to the PharmD program. And then towards the end of the PharmD program, that's where the interactions with UNC began. Getting ready for mid-year at the end of my PharmD program. Back then, you didn't use the internet to find out about fellowships. You saw it in like the back of a journal. And then you went to mid-year and put your resume into a slot that someone pulled out and said, oh, you might be suitable for the fellowship that we have. And that's where I met Dr. Brower. So that was in, I guess it was in 1991. That's when I met Dr. Brower at mid-year and really decided that it was the best fit for me. It sounded really exciting. I wasn't sure what I would do with it in the end, but the scientific work that she was working on and the fact that I had a chance to work in both academia and industry, it opened multiple paths. And I think that's really important, not to shut any doors. So I was opening two possible doors at that time. So then I came to UNC. I did the UNC Burroughs Wellcome Drug Development Fellowship. And without doing that fellowship, I would not have been able to go to FDA and work in the Office of Clinical Pharmacology. In our Office of Clinical Pharmacology, we have a mix of PharmDs, PhDs, PharmD PhDs. We don't have any PharmDs who don't have postdoctoral training. So I really needed to do a fellowship in order to have the qualification. So that really set me up, even though at the time I didn't know what I was setting myself up for. And that is part of my message. Follow a path where you're interested in what you're following, even if you don't know what the end result is going to be. So I worked with Dr. Brower, and then I also worked with Dr. Ginny Smith, who at the time was at Burroughs Wellcome, which doesn't exist now because that's what happens with drug companies. But at the time, it was Burroughs Wellcome. And following my time at doing the fellowship, that's when I went to FDA, and that was in the summer of 1994. And I started out as the clinical pharmacology reviewer. I enjoyed what I was doing so much that I wanted to expand work beyond the portfolio of just one person. So I became a team leader. And that meant that I got to look at all of the work that was going on in antiviral drug development, which if you think about the time that I'm talking about, the late 90s, it was a very exciting time in antiviral drug development, which it is again now. But who knew at that time how things were going to change? And then I became a deputy director of the division, and I became the division director of course, the beginning of 2020, not knowing what I was in for. So that's the journey I went through. And so part of my message here is, different people have different approaches to goal setting. Some individuals have a very specific goal that they want to follow. So my goal was it was less specific. I knew the type of work that I wanted to do, and I continued going from place to place, feeling like it was right for me and interacting with individuals that I really connected with and that I could learn from. So for me, that worked out. I'm not saying that it's not okay to set a very specific goal and follow that, but this was a path that really worked for me. So next, I'm just gonna give you a little bit of information about FDA because I know it's a big black box. So most people, when they think about FDA, they think about drugs, they think about food, because both of those are in the title of the name, Food and Drug Administration. So the FDA itself has about 18,000 employees, and I work specifically in the Center for Drugs. So that's the center where all of the therapeutics are reviewed. So drugs and then therapeutic biologics like monoclonal antibodies. We don't cover vaccines. So that is one difference. That's in the Center for Biologics. Within the Center for Drug Evaluation, there are a lot of other offices. There are a lot of offices, and I'm in a sub-office of the Office of Translational Science. So I'm in the Office of Clinical Pharmacology, and that's mostly what I'll be talking about this evening. It's about the Office of Clinical Pharmacology. But just to let you know, there are other offices that do other work. OGD is the Office of Generic Drugs. That's where all of the generic drugs are reviewed. OND, Office of New Drugs, that's where all the physicians are that review the new drugs, and we interact with them every day. And the Office of Clinical Pharmacology has about 270 employees. So now I'm going to focus on the Office of Clinical Pharmacology, which is where I work, and I want to focus on why we do what we do. And we are an office that has a vision and a mission. I know UNC School of Pharmacy has a strategic plan, a vision, and a mission. Some organizations put their strategic plan together and then it's put aside, and it doesn't really matter. It was just an activity you did, and then you come back 4 years later when it's time to work on the next 5-year plan, and you have no idea what you achieved from the previous strategic plan. So like UNC School of Pharmacy, our Office of Clinical Pharmacology really does use their strategic plan. And we just redid the strategic plan. And when we did that, we did not change the vision and the mission. So the specific work has changed over the past 5 years, but the purpose of what we do has not changed. And I think that's one reason that I've really enjoyed working there so long. There's been a very consistent vision and mission of the work that we do. And I'm not going to read this because it's a lot of small print. I assume you'll have the slides afterwards. But overall, putting it together, we want to enhance drug development, promote regulatory science and innovation, so we're not just doing reviews. And in the end, we want to inform the optimal use of medications. We want to make sure patients receive the right medications and that they receive instructions so they know how to use them appropriately. And that's pretty much the focus of the work that we do every day. There's specific questions that we ask during drug review and also during all of the other regulatory research and policy work that we do. And these are the 4 main questions that we ask. We want to make sure that there's clinical pharmacology information that can help support whether or not a drug actually works. So it's not just the big clinical trials that inform that. There's also clinical pharmacology information that we're able to review and determine whether or not the drug works. Very important is the dosing regimen that's going to be approved and then prescribed and then taken by the patient. Is it the right dosing regimen in general? And then are there any specific reasons that those doses might need to be changed? Is it needed because of impaired renal function, impaired hepatic function? Do women and men need different doses? Are there different doses for different ages or different races? And then are there clinically relevant food interactions or drug interactions? All of us receive, the pill bottles telling you take it with food, take it without food, make sure you're not taking with this other medication. And that's the work that we do specifically within our office. This is very related to precision medicine, which I know UNC has been quite involved with. So although there's a lot of different things that we do, in the end, it all informs these 4 questions and how we evaluate drugs and think about these 4 questions. Now I want to point out the impact that I have seen over the past 30 years. And so I will highlight the response to HIV/AIDS, because that's what I've had the most experience with, and I've been somehow involved with that for my entire career. So this is before I joined FDA, so this is what had happened before I got there. The AIDS epidemic began in 1981, and clinical trials with the WD, which in this area, started in 1985, and then it was approved in 1987. And that's about where we were when I joined FDA. When I joined FDA, there were 4 drugs that were approved for treatment of HIV. And the HIV epidemic is one of the reasons that FDA came up with the accelerated approval program that is now used quite a bit for oncology, also for some of the neurology drugs. So it really did drive that. And sometimes you think that the government can't make new rules; they're stuck in their old ways, and this is how you do things. And when you send a drug in, it's going to take 10 years to get it approved. But this shows that that's not true. And the HIV community was really behind this and pushing this. They were not going to stand for drugs taking forever to be approved because they were dying. So we did listen. There were a lot of listening sessions with HIV patients and their families, and things were changed. And it has worked since then. And 1992, that's when combination drug therapy began, again, located in this area. It was 3-TZ instead of OVD. So again, in the North Carolina area, that's where that was. And a real turning point was when the first protease inhibitor, sequinavir, was approved in 1995. And that was the beginning of what was called, at that time, highly active antiretroviral treatment. Indinavir and ritonavir were approved. This is when all the drug interactions started to be really highlighted. And for me, this was probably one of the more exciting times in my career. We were very, very busy. We were probably working about 16, 18 hours a day trying to get the drug applications approved. But it was worth it because we were so excited and we could see the impact that we were having. However, the drugs were very difficult to take for the patients. Many of them had food interactions, drug interactions. You might be taking 2 different drugs that had different food interactions. So it was very, very difficult. So I'll skip over 2003 for just a minute. I'll point out 2006. That's when the first once daily single tablet regimen was approved. So that was, again, a very big turning point where it became much easier to take the HIV regimens. One thing that I want to point out that most of you probably have not heard of is the President's Emergency Plan for AIDS Relief, and called PEPFAR. And this is a program through which the United States government helps other countries who don't have as many resources prevent and treat HIV. And it started in 2003. It was announced at the President's State of the Union. And it's one of those things you kind of hear it and it goes in one ear, out another ear, like, what's going to happen with this? And it has, at this point, saved about 55 million lives. About 5.5 million babies have been born not HIV infected because their mothers took HIV therapy. So it's been tremendously successful. And our office has been very involved with this. But through this program, the US government purchases cheaper HIV drugs, so drugs that aren't approved for use in the United States. It's a lot of generic drug companies from outside the US, but we have to review all of them. Because in order for the US government to purchase the products, we have to make sure that they are of the quality that we would sell them in the United States. And this has been one of the most exciting things that I have worked on being at FDA. So where are we now as far as HIV? There's been a lot of progress. Treatment as prevention, individuals who have their viral load undetectable, for the most part, there's reduced transmission of HIV. We now have pre-exposure prophylaxis, oral therapy, and there's also an injection that can be, it can be injected every 8 weeks for prevention of HIV. And now, very exciting statistic, HIV life expectancy in the US now matches the general population if therapy starts when the immune system is still fairly intact. And that is such a change from the time when I joined FDA. I mean, I remember going to advisory committee meetings where patients who were almost dying were just begging for drugs to be approved. Unfortunately, many of them did die soon after that, but some of them are still alive now because just in time that the products were approved. And it is so exciting to see them at later advisory committee meetings advocating for future patients. But there's still more work that is needed, of course. There's a lot of HIV cure research going on, a lot of it through UNC. So I'm always very excited to hear about that. HIV, a pre-exposure prophylaxis for all relevant populations, that's not completely available now. And work that we do continue to do at FDA to make sure that we're meeting the needs of the patients is continued discussions with the HIV community. And our most recent one, I think, was about a month ago. And so we had a very good relationship with them. It's not just Office of Clinical Pharmacology, also the Division of Antiviral Products, which is in the Office of New Drugs. Very good relationship with many of the different organizations of the HIV advocates. And they let us know what they think, what we're doing well, what we're not doing well, what we need to do more of. And most recently, it was interesting how things have changed, that we need more studies of HIV drugs in older patients. Early on, that was not relevant, unfortunately, because no one was living very long with HIV. But now there's interest in what happens when someone takes these drugs for 30 years? Do we know? What happens when it's more frail patients because they've aged so much? And we don't know all of that yet. Just the fact that these new questions are coming up. There are questions about the transgender population. We have to make sure that we're looking at all of the relevant subpopulations. So we do continue to have discussions with them. I think there's another one scheduled in about a month or so. And it is really exciting to interact with the specific patients that we are affecting. But we're not all about HIV and AIDS. There are other, even within the division of infectious disease pharmacology, there are other areas that we work in. So another one that's very exciting is hepatitis C. Before 2011, therapy for hepatitis C lasted for 24 to 48 weeks. It typically involved ribavirin and interferon, which was barely tolerable. Many individuals had to have their dose decreased. They would have to go off therapy. And the treatment success, even if they were able to complete treatment, was less than 50%. The first direct-acting antiviral product was approved in 2011 for hepatitis C. And now hepatitis C is almost completely eliminated, pretty much curable, with greater than 90%, 95% treatment success, to a point where we barely do any HCV drug development work within our division now. I recently had a data call that I had to respond to, like how many hours has your division spent on HCV drug development over this certain period of time? Answering the data call actually, I think, took me longer than the work that we had done, because we've done so well. Of course, there's always more that can be done. There's certain subpopulations that may not be reached yet, particularly pediatrics. But it's such a difference over the past decade. Ebola virus, we have done work in that area also in 2013, 2014, when there was the outbreak. There were no targeted antiviral drugs, really nothing even with animal data. So there was this huge outbreak. There was this outcry for, well, what do we give these patients? We have to do something. So there were some drugs that, well, maybe they might work in Ebola. We studied maybe this other similar virus, or there was some in vitro work, maybe a little bit of animal work, but there was really nothing with like true evidence that it worked. So we were on calls with NIH and CDC almost daily. You're trying to figure out how can we possibly extrapolate the little bit of information that we have to patients, because there was no choice. And that did lead to 2 monoclonal antibody products being approved for treatment of Ebola virus in 2020. And that's because there was a combination of responding quickly, but to the best of our ability in a scientific way, and collecting as much information as possible. So this was 2013, 2014. You can imagine the scenario did come up again less than a decade later. And then there are products that are indications where we can't study it in humans. For example, anthrax and plague. We can't do clinical trials for anthrax and plague. Thank goodness. But we do need to study it and be prepared. And in order to do that, there is legislation and a pathway called the animal rule, which means a product can be studied in an animal model that has been verified, and see that it works in animals, and then we can link the dosing to humans through pharmacokinetic data. Obviously, the Office of Clinical Pharmacology is very involved with that. So products have been approved for treatment of anthrax and plague and for prevention based on the animal rule. And then COVID. So just like I mentioned for Ebola, we were in a situation where we needed to meet an urgent public health need that we were hearing about every hour on TV. But we need to make a thorough evidence-based decision. And it was a very difficult time. Looking back, maybe it was exciting. I actually don't remember the first 6 months of COVID at all. We were so busy. But in the back of our mind, we had to keep the mantra of clinical pharmacology in mind, that each patient needs the right drug, they need it at the right time and the right dose. So while we wanted to treat people as quickly as possible, and there were all sorts of stories out about drugs that may work, so you need to immediately give them to the patient. We're never going to know whether the drugs really work if they're not studied appropriately. So that really was a struggle, just like it was with Ebola, but this was on a worldwide scale. And so we were very involved with that. And I want to talk about one specific drug, Paxlovid, which I'm sure all of you have heard of, Paxlovid. So we were involved throughout the drug development of Paxlovid. And as it got to the end, well, not really the end, but it got to the point where it was going to be authorized for emergency use, it was clear that it's really not that easy a drug to prescribe to patients and to give appropriate instructions. And also, it was going to be given by a wide range of care providers, including pharmacists were also going to be able to prescribe Paxlovid. So we had to make sure that individuals who weren't going to have a lot of time in the clinic with the patients, who weren't used to prescribing this type of medication, had all of the information that they needed so they could make it clear that they had thought about whether this drug is appropriate for the patients and that the patients had the appropriate instructions. So there was a checklist that was put together. So the drug company put it together and the FDA worked with them to make sure that the appropriate information was on it. The topics it addressed were the test results, do they have the appropriate risk factors for Paxlovid, which includes information about the symptoms, the symptom onset. So there were very specific criteria of whether it was appropriate for the patient population. And then most difficult really, and then most related to clinical pharmacology was the drug interactions. So there was a page-long list of potential drug interaction with Paxlovid. So we were tasked with pretty much verifying every single drug on the list. So does this need to be on the list? Sometimes it's very easy to say, well, something bad might happen, so just don't give Paxlovid if the patient is on this drug. But then that deprives them of therapy. So we wanted to make sure we weren't doing that. So that was a lot of work. We were used to it from the HIV epidemic, but we had to remember it was going to be different prescribers this time. The HIV community was very used to prescribing ritonavir and thinking about drug interactions. But now there was going to be a whole different group of individuals prescribing it. It was a little bit different setting because you have to start the drug so soon. With HIV, you have a little bit longer before you immediately need to start the product. But with Paxlovid, you really need to start it as soon as possible. So we put together a color-coded list with red, don't give together, yellow, there's some things to think about. And then maybe you can give them together, and then green, everything is okay. And of course, no list is ever complete. So then communicating the fact that, well, it's as complete as possible, but it may not be complete. So these are the type of activities that we've been involved with during the COVID pandemic. So at the FDA, behind the scenes, there's much more going on than just drug review, though, and that's what makes the job so exciting. Everything we do does look forward to drug review and drug development, but there are many other things that we do. And I think that that's one thing that's fairly unique within our office is, although it's not a requirement that we work on policy and research, it's definitely encouraged. So pretty much everyone works on extra projects beyond just drug review and working on drug development. There's a lot of work on policy, so coming up with guidance documents that informs drug industry of how they should develop their drugs. And then we need to follow the guidances also. If we ask a company to do something, we need to respect that when they've done that. And then we do a lot of research. And much of the research does lead to presentations and publications. So some policy examples, the types of guidances that we've been involved with, specific populations such as pediatrics, women, pregnant women, novel drug products, all of the pharmacokinetic guidances come out of our office. And then there's safety and quantitative medicine, such as population PK, exposure response. There's a lot of applied regulatory research that goes on. Some individuals are surprised sometimes to hear that we do actually some research within our office. There's a lot of research around COVID, and we look at patient factors such as diversity of populations, precision medicine. We do safety-related research, model-informed drug development. Not huge clinical trials. There are some smaller clinical trials that we can partner with other organizations, but then we do a lot of in vitro work, a lot of animal work. And then we also do survey type work. I mean, we see more data than anyone else for the drugs that are developed in the United States. So we can go back into applications and look across all applications and like what happens with renal impairment across all drugs that were approved for the past 20 years. We have that data at our fingertips. So that type of research is also done. And then a lot of communications. Much of it is based on the research that we do. And we also do review summaries and workshop summaries. And most individuals in our office really do appreciate the fact that they get to publish and they get to have their names on publications. It's very rewarding. And then we also do external presentations. It might be academic settings, such as this. It may be at professional meetings. Many of us are involved with American Society for Clinical Pharmacology and Therapeutics, American College of Clinical Pharmacology, and some of the pharmacometrics organizations, or even some of the disease-specific organizations. Within my division, we do have individuals who present at infectious disease and virology meetings also. And then FDA sponsors workshops and webinars also. I mean, I was complaining recently that my calendar was just overwhelming because there was an FDA-sponsored workshop that seemed very relevant to my work, like every 3 weeks. It's like, how am I possibly supposed to do my regular work if there are all these fantastic workshops to attend? Luckily, they're recorded. So the ones that I missed, I can go back and look at them. And we also have many collaborations. For example, I'm sure you're all familiar with CIRSE at this point, the Centers for Excellence in Regulatory Science and Innovation. So I was very excited when the UNC Duke Research Triangle CIRSE was funded. And it's a big deal. I mean, it's a huge, huge deal. And I'm looking forward to our office working with you on this. And we also, other collaborations, international harmonization. So drug development is international, it's global. And if the United States asks drug companies to do something, and then Europe asks them to do something else, and then Japan asks them to do something else, that's very difficult, and it's not the best use of resources. So ICH is an international harmonization group that gets all the regulators together along with industry and works on collaborative guidances. I've been involved with the drug interaction process over the past 5 years. So we're coming up with a drug interaction guideline that will combine the US, Europe, and Japan so that all of the expectations are the same. So it's very time consuming, but it's very exciting, and the result hopefully will be worth it. And then we have memoranda of understanding with other organizations, including some academic institutions. And those are very specific to whatever the needs are of the two organizations. So now I'm going to step back and talk a little more broadly about the components of your script or my script as you're looking forward to your career, and the components to think about and which really build on each other. I mean, any career, there are basic skills that you need to have, and that's where you start. So showing that you have the basic skills, that's kind of how you get in the door. And so, it's important to think about, well, what skills do I really want to build? Is it worth it to me? Will I get something out of it? And then as you move along, there are advanced skills that you need as you want to move through the organization. It may be more scientific skills to move in a scientific pathway, or you may want to take more of a leadership path. But the advanced skills that really align with how you want to proceed. And then you have to be organized. You need habits and processes. You can have all the ideas and all the skills, but if you don't put them together and have processes that you follow and make sure that you have good habits, then it's not going to lead to anything. And communication is very important at FDA, as it would be at any other organization, both written and oral. Critical thinking, so how do you apply these skills, the basic skills and the advanced skills? And then the soft skills. The term soft skills, I know some people don't like it because soft skills are hard. They're difficult, but the interpersonal communications. And I mean, these are skills that we see as very important within our office. And we do train on all of them. So that's something that I really like about working at FDA, is that we have set things up so that if someone comes in with maybe 3 of these pretty well developed, we will work with them on the others. And it's very important, I think, to find a workplace where if you have some skills and you need to build others, that you're going to be supported. And I've been supported throughout my career, and now it's my privilege to be able to support others. And then editing the script, a work in progress. I mean, I'm coming probably towards the end of my career. I mean, maybe I have another 10 years or so, but things can change over time. And it's important to pause every once in a while, think, look back. How are things going? What is really important to me? What is essential to me? And is that what I have been working on? And things to think about at the beginning of your career. What activities cause stress? Those might be activities that you don't want to have a career that you would be involved with. For example, my bright idea of being a physician when I was younger, having to be like on the spot making that type of healthcare decision, that would not be me. So that would be a stressful job, and that would not be the appropriate job for me. What activities relieve stress? It's nice to have a job where like some of the things that you do actually decrease your stress. So many of the articles that I read and just taking in information, I don't have to actively do something, but it's part of my job to learn. And to me, that relieves stress. Or some of the interactions I have with individuals in my division and the whole office, those relieve stress. So it's good to have part of your workday where you can enjoy it. And again, think about activities that you enjoy. I was always like the geeky kid who liked math. It embarrasses my kids when I tell them that I used to do my math homework for fun. But that's the way it is. And it worked out okay. So it's important to think about this at the beginning of your career, but also as you think about maybe shifting to a different position. I have had some different opportunities to shift just a little bit. Sometimes they worked out, sometimes they didn't. But in the end, I think I've ended up where I really did belong. And that's why the job has been so rewarding. And then within your job, look for internal consistency. After you've thought about what is really important to you, look at the priorities of your organization. As I mentioned, we did do a strategic planning exercise we started about 2 years ago. The mission and vision stayed the same, but we came up with new priorities. And the 3 priorities are advance our science for the benefit of patients. So that's the science part. Bolster patient-centric engagement. So making sure the work that we're doing really does benefit the patients. And then we get feedback from them so that we know that we are benefiting them. And then elevate our people. So that's how we mentor each other. That's how we make sure that there's career development available for individuals. So I can look at this and ask, is this consistent with my values, my personality, and my work style? And I can say yes. And I think that's why I have enjoyed my job so much. So these are my closing thoughts. Many of my presentations, I do like to have a quote from a book. So this is from the book, What the Eyes Don't See, a story of crisis, resistance, and hope in an American city. So it is about the Flint, Michigan lead in the water crisis. And Dr. Mona Hanna-Attisha was the individual who was probably the most responsible for turning this around. And her book is excellent. So I highly recommend it. And so I think this quote really captures what we do at FDA and in the Office of Clinical Pharmacology. So it's about people and community. That's what science is supposed to be about, not an academic exercise for the ivory tower or racking up publications, grants, and offers of tenure. It's about using the tools and technology available to make lives better. So you might get the publications, the grants, and the offers of tenure. Those are important, but that's not the end goal. That gets you to the end goal. So that's how I would like to end my talk today. I do have acknowledgements. So I didn't put specific names because there's so many people, I didn't want to miss anyone. But many individuals within the division I work in, the 2 clinical divisions I work with, anti-infectives and antivirals. The strategic communications team made the nicer slides that you see. So my PowerPoint skills aren't that great. So there are a few that someone else did. And then VCU School of Pharmacy, where I got my initial pharmacy education, and then the fellowship at UNC, clearly invaluable. And then also ASCPT. So I did miss talking about ASCPT. And so part of the collaborations that we do have individually and as an office is working with some of the societies. And so I've worked very closely with ASCPT. I've been on the board. I was president from 2017 to 2018, which is another type of opportunity that is really encouraged within our office. So if you want to learn more about what we do in the Office of Clinical Pharmacology, the link to our annual report is here. You can also just Google 2022 Office of Clinical Pharmacology annual report, and it's about 34 pages long, and that's just a summary. And I'd be happy to answer any questions about my experiences or anything else about what's going on in the Office of Clinical Pharmacology.
Thank you, Kellie, so much for your work and also for your words here, for your inspiration. I love the end quote about making people's lives better. We say here at the pharmacy school, everything we do begins and ends with the patient in mind. So that really resonates. We're going to open the floor for questions for Dr. Schoolar-Reynolds. And for those of you on Zoom, we also have the chat being monitored. So if you have a question, please put it in the chat and we'll get that one answered. I might start with one though. There's a couple things that resonated with me for your vision and mission, and it included the words innovation and also patient individualization and patient-centric at the very end. Can you talk about the use of generative AI and how that will overlap with the work that your office is doing from an innovation side and also from patient individualization side?
So it's early.
Yeah,
and
for sure.
So I'll speak more about what's going on within our office.
Yeah.
And when we are looking for the best way that we can use AI and generative AI to help us with our review products. So there are a lot of things that we do routinely. We have like specific data sets that come in. Are there ways that some of that work, the initial review, to be done using AI? And we have an AI group within our office that is working on that. So that's one thing that we're looking at there. Obviously, I mean, in the end, we're responsible for the product. That's what I have the most familiarity with.
Okay. Yeah. Great. Yes, Dr. Kohn.
First of all, thank you very much for the lecture, and your work has been so impactful. The number of people your lives have affected us it's beyond even comprehension. But I have a question. In the years that you've been at the FDA, what is the general trend in the size of phase 3 clinical trials, given the advent of biomarkers, given the advent of modeling, but also, on the other hand, the need for genetic information and a different population, people in the population group, and the need to know more information. So do phase 3 trials have gotten bigger, smaller, shorter period of time, longer period of times?
I mean, it's variable across indications. And one thing that at this point is hard to replace is safety information. So, the safety, so gathering safety information from enough patients, thinking about how many will be treated in the end. So that hasn't really changed. Now, as far as, the size of the clinical trials, where we're getting the specific efficacy information, I mean, there are more areas where we're comfortable with biomarkers and accelerated approval, although typically they're larger trials that are done afterwards in order to verify that. Now, the HIV clinical trials have gotten smaller because we've gotten more comfortable with the surrogate markers. So it's probably trending for the most part slightly smaller and faster, but not a lot smaller. Maybe now the time to get to the phase 3 clinical trials is probably where there's more of a difference. So the actual drug development time, able to provide quicker information on how to better develop the drugs, that's probably shorter.
Mary?
Yep. Thank you, Kellie. It was really informative to see the behind the scenes look at how you're structured and what you do. And I'm on faculty here at the school. But I'm particularly intrigued by the comments you made around all of the skills that you develop within your workforce and your team. And you mentioned that when individuals come into the organization, that you have training for those who need it in certain areas of those soft skills, whether it's communication or habits and processes. Do you know that when they come in, or is this behaviors that you have experience with over time and you identify that people need certain training and then you provide it? I'm just curious how you go about with that.
It's both.
You're trying to do a lot of that here.
It's both. And it depends on the experience the individual has. And there's certain expectations we would have for someone who's been working in industry for 10 years. We do have some individuals who come to us from industry. So if someone's been in industry for 10 years, our expectations, for one thing about their level of maturity, is different than someone who is just graduating. So we do think about that. We have no expectation of any regulatory knowledge. So our baseline there is zero, unless someone's been working in industry. But individuals coming out of PhD, sometimes they're a little bit nervous when they come for interviews. Like, well, we don't expect you to know how to do drug development. We can teach you how to do drug development. We do expect the basic clinical pharmacology and math skills. So because for one thing, why would they be applying for the job if they didn't have those skills? Although clinical pharmacology is such a broad area, sometimes there is a misunderstanding of what we actually do. So we make sure they look at some of our work products in advance. So for someone coming straight out of a PhD program, or someone with a PharmD who's coming out of maybe a 1 or 2 year fellowship, we expect them to have the basic skills, a few of the advanced scientific skills, and decent communication. If they have stellar communication, it's great. But decent communication, ability to interact in a small group. We also have them do a seminar, so we see how they handle a crowd. But I mean, things like that, I don't even know if they're semi-quantitative. They're fairly subjective. And in the end, you never really know. and that's why we do have the ability to train, and we work with individuals very closely during their first year. We get their individual development plan in place within about 3 months. And we work very closely with them. There's a team leader who's working with them, and we get feedback. And because we don't want things to go on for a year and have them develop bad habits.
Great lecture. Really, really enjoyed it, particularly your path in the beginning where you talked about not really having a plan, but just going down the path of what you enjoyed and kind of shutting other doors, but leaving others open. I thought that was really great advice and just love the lecture overall. My question for you, as you were speaking, I was glad you got to the part about the original research that you guys do. I'd love to hear an example of maybe one of your favorite research projects that you've been involved in, to just give us a feel for that, other than you mentioned the renal failure, kind of a data mining approach, but is there another project you could give us an example of to give us a feel for that?
Some of the research that's been done by individuals in my division, well, there's one individual who is looking at the various ways that hepatic impairment can be categorized, which is very difficult. The hepatic impairment studies looking at mild, moderate, severe, and the effect on pharmacokinetics. There are various different ways you can categorize. The Child-Pugh is the most common. There's the MELD score. There are other ways. So we did have one individual who went back through as many drug applications as possible, looked at how it was categorized and the effect on pharmacokinetics, so the relationship, and then calculated all the other measures to see which ones would be the best. We're still coming out with Child-Pugh, which, I mean, the liver's tricky. And the problem is it depends on the characteristics of the molecule also. But that's the type of work we're able to do. Go back and look at all the renal impairment studies that were done. Is it even valuable to look at mild renal impairment? Is there only a certain time where it's valuable? We look at the types of studies that are done by companies. The area that I've been most involved with is drug interactions. So how does in vitro work predict the clinical results, and looking at different specific in vitro results? Where can we do different cutoffs that you need to do a clinical drug interaction study if this is your in vitro result? And then moving the cutoffs. I've been very involved with that also.
Yes.
Previously, you mentioned the use of incorporating, further specialized populations within the use of testing for PrEP and PEP, as well as any other antiviral drugs. What have you seen in your experience within kind of your office regarding similar studies for aging populations as well as pediatrics regarding gender-affirming care and HRT?
Not a lot yet. Actually, it is being talked about within the division of antivirals. And that is also one of the topics that's come up with the HIV community. It's important that we continue to include that, but it's very early.
We actually have Mackenzie Cottrell, who works in my laboratory, is very interested in this issue and just presented some data at the HIV clinical pharmacology meeting in September in Rome, looking at intracellular metabolites of PrEP and hormonal therapy, comparing cis and trans gender populations. And it doesn't look like, with hormonal therapy at least, that there is any sort of issue related to changes in PrEP efficacy, at least with Truvada and also with Descovy, TAF-containing products. So a little bit of information, although it's challenging to get to. It's challenging to find patients to enroll in clinical trials, but at least there's some promising information in that space. Yes.
Hi, I'm Sophia Stein. I'm a first year in the PharmD program, and I want to say thank you so much for speaking to us today. This was a really informative lecture, and I appreciate that. You talk about in your strategies that one of your priorities is bolstering patient-centered care, and I was wondering if you could expand upon some of the they're using to elevate that.
So in the patient-centered care, one thing that we're working on is like the language of information for patients and making sure that it's clear to patients, and also that information and product labeling that's for the healthcare provider really provides them with the information that they need to communicate to their patients. I mean, we all have seen the package inserts that are really, really long. They're very scientifically dense. And you're trying to balance that with providing the information that the individuals who want a lot of information can have, also with making it clear what the most important information is. So that's one area that we've been working on. A lot of work around providing usable drug interaction information, looking at dosing instructions. Sometimes the instructions, particularly maybe related to food, I don't even understand them sometimes. And you're making sure not only is it understandable, that it's actually doable. If someone's taking a medication for 5 days, it's not that big a deal. But if you're going to take something for weeks and weeks at a time, and the really strict instructions about how to take it with food, it's very disruptive. And sometimes it's because it's just going to be required because of the characteristics of the drug. But sometimes it's because maybe it could have been studied another way and we would have better information, and we're just trying to be conservative and make sure that the drug does work. So that's the type of work that we're doing, some of it. We also have, we do have patient engagement sessions also. There are specific meetings that we've had with patient communities. And then also at some of the professional meetings like ASCPT, there's often a patient forum where patients with a specific disease, they come in and talk about that experience. This year at ASCPT, there were patients with long COVID, and our office director, Isam Zaneh, was part of that also.
Dean White, did you have a question?
Thank you for the wonderful presentation. Really enjoyed it. I've been seeing a lot of buzz around the FDA, taking steps to increase diversity and ethnicity in clinical trials. And I saw that you included in your presentation how important that was. Can you share some of the strategies that the FDA has been taking to include representation in clinical trials? And is there a baseline where there has to be a certain percentage of certain groups in certain studies? I don't know if it's that specific.
There's not a baseline, so I can answer that. There's really not a cutoff, and there's nothing that really allows us to require a certain representation. One thing that we do specifically within our office is make sure there aren't barriers to including a diverse population, so that the studies in renal impairment, hepatic impairment are done early so that patients can be included in clinical trials, that we have drug interaction information early so that in phase 3, patients who are on certain medications aren't excluded. So that's one of the big roles of clinical pharmacology. When phase 3 clinical trial protocols come in, what we really need to do is look at the inclusion and exclusion criteria. Does it look like it was just cut and pasted from every other phase 3 study that was done? Or is there a specific reason that a BMI greater than 30 is an exclusion criteria for no reason whatsoever? And we are starting to, so one of the workshops that our office co-sponsored was one on including obese individuals in drug development, because they're often left out. The point was made that it's 40% of the population. For anti-infective drugs in particular, it can be a problem with them not getting high enough drug concentration. Or if it's a fat-soluble drug, maybe the drug will be in their body for too long. So we are starting to look at that. And I've seen a lot in the lay press recently about that workshop, even though it was about a year ago. So that's also something that we're looking at. But our specific job, like within our office, is to make sure that the barriers are decreased for increasing diversity.
Dr. Schoolar-Reynolds, thank you so much for your presentation. We learned a ton from you, and so thank you for spending time with us today.
And thank you for all the questions.
Yeah, the questions are great. Thank you so much to Hal and Carol for your support of this lectureship. I'd like to thank you all for your attendance and for your questions. They're terrific. And I encourage you all to move upstairs to the second floor lobby, where there'll be refreshments and some time that you can spend with Dr. Schoolar-Reynolds for additional fellowships. So thank you all.